Everybody's kind of fading there at the end. I just thought if I start talking about oxidation of unsaturated fatty acids, I'm going to lose everybody. How are you guys feeling about this exam? Unprepared or prepared? Unprepared. A lot of material? Lots of it, yeah. There's a lot of stuff to memorize here, yeah. Yeah, a lot of things. Yes, well, I hope it's logical, because I think the more logical something is, the more you can learn by thinking instead of learn by memorizing. One of the reasons I emphasize the respiratory control, which of course won't be on this exam, but it will be on the final, is that you can think your way through it. And knowing what we've talked about with respect to electron transport and so forth, you can see those connections. I mean, you can see why, if I add 2,4-DNP, why it is that cells burn energy. You can see why it is that it was considered a miracle diet drug, for example. Okay? So I think those things, like I said, they're logical when you see the interconnectedness of that. But it means you do have to understand the electron transport system. If you don't understand the electron transport system or know how that operates, then nothing else is going to matter. So you have to understand that for sure. Okay, I'll shut up and let you guys ask questions. So, what do you want to know? Okay, is there anything on this test that's going to be from the first test? The answer is no. Okay, so I don't go back and pick up things that I've talked about before. The final exam will have that, but this exam will not.
I'm having some issues still with the
With the what?
phosphorylate regulation, where you have
You're talking about the glycogen phosphorylase?
it's like the inactivated forms and the activated forms.
All right, so let's go back and take a look. So if we talk about this,
Yes.
is that what you're talking about? Okay. So, like I said, I think it's easy to look at this and say, oh my God, there's so much stuff here. But in reality, moving left and right involves phosphate on or phosphate off. Okay?
Why would we ever need to want to
Okay, so his question is, why would we want to go phosphate on or phosphate off? Okay? That's controlled by hormones. That gives us hormonal control. So if you are, in that emergency situation, then hormonal control is helping you to get out of it, because it's phosphorylating your enzymes, phosphorylating these enzymes, to allow the body to produce a ton of glucose that's going to get you out of the emergency situation. If you don't have that hormonal control, you can be as scared as you want, but it's not going to make any difference because you're not going to produce any more glucose. Does that make sense? Right. Right. So that's the situation where you have the child under the car and you can literally pick up the car and save them. That's what that enables you to do. Okay? So hormonal control is essential for an animal to be able to immediately or almost immediately respond to something. So going left and right gives hormonal control. Right? That hormonal control goes two ways. As we move to the right, we're making the enzymes more active, meaning that there's more glycogen that's broken down, which means there's more glucose being made, and the muscles have a ton of energy. As we move to the left, the enzymes are being made, in general, less active. I got to sneeze. I hate it when that happens. It'll come out in a minute. It always does. Being made less active, and that's important. Dephosphorylation happens as a result of stimulation by insulin. Going to the right with epinephrine, going to the left with insulin. It doesn't matter whether it's top or bottom. Both of those are going either phosphate on or phosphate off. So as we go to the left, phosphate goes off. As we go to the right, phosphate goes on. That phosphate goes on to either form, the R form or the T form. As we go up and down, we're talking about allosteric regulation. That's where molecules can influence things. Now, it turns out molecules influencing things happens instantaneously. Hormones travel at the speed of your blood. That takes a few seconds. Instantaneous means constant. Constant monitoring of these guys with respect to being on or off, that is, being an R or T form. That turns out to be really important, again, in muscle. If I take off on a sprint, immediately my ATP levels fall. Immediately that happens. When my ATP levels fall, okay, you see I'm not going to go to T, I'm actually going to go to R. And R is going to be more active. So immediately my muscles don't even have to wait for the hormone, they're on. And they start breaking down glycogen almost instantaneously before the hormone even gets there. Then when the hormone kicks in, bam, I've got another dose, right? So what this gives me is an instantaneous control up and down, and it gives me a slightly slower control left and right, both of which are important for me to be able to manage my sugar in the body. Does that make sense? Okay, so his question really, I think your question has to do then with, why does hormonal control play into this? Because if I've got instantaneous control, why do I need hormone control?
Well, I understand why we need hormonal control. I just don't understand the hormonal control in the inactive versus the active.
Okay. Well, let's say that I am in that grizzly bear situation where the grizzly bear is chasing me, right? So when I start that situation, I've got a certain percentage of my enzyme in this form and a certain, let's say, 30% and 70%. Okay? So my hormones hit, epinephrine hits, adrenaline hits, and this gets converted into this, and this gets converted into this, right? So I've got 30% and I've got 70%, right? Only instantaneously. Because do I have glucose in my cells, in my muscle cells, when I'm running away from the grizzly bear? No. So if I don't have glucose when I'm running away from the grizzly bear, T is going to R. So converting this guy to this guy enabled it to go to R almost instantaneously. That's why we say this form is more active than this form is. Because if I'm running away from a grizzly bear, there's nothing left in the T form here. It's almost like a diagonal. That's right. It's almost like a diagonal. Yep. Now, you might say, why the complexity? The complexity gives the body the ability to respond to things in very different ways. Okay? We're not always in a flight or fright situation. You're not in one right now unless you're afraid of the exam, right? Maybe some of you are afraid of the exam. Maybe some of you are afraid of the exam. Okay? But your body still has to be able to respond to the situation that you find yourself in right now. That sort of basal level of things bouncing around, managing that right now, is what's going on in the up and down.
Okay, so I understand the reason going this way. Why would we ever need to go from, like, phosphorylase A
Back to B.
to the inactive T form?
Okay. Well, how do we get back over here? All right? So how would I get up to here? I'm running away from the grizzly bear, and the grizzly bear stops chasing me. My liver, which has been dumping glucose into the bloodstream, keeps dumping glucose into the bloodstream, but my muscle cells are no longer using it, right? So what happens to my blood glucose levels, my muscle glucose levels? They go up. Do I want to be breaking down glycogen when I have glucose? I don't.
So you need to phosphorylate in inactive form, but why would we need to go back to the unphosphorylate?
Okay. Glucose is a poison. So when glucose concentrations are too high in the body makes insulin to take the blood glucose levels down lower. The cell takes up glucose. Okay? If it's taking up glucose, it doesn't want to be breaking down glycogen. So insulin stimulates the dephosphorylation of the enzyme. These are very good questions. You're asking exactly what you need to be thinking about with this. So insulin's going to favor the dephosphorylation of this enzyme, and the dephosphorylation of this enzyme is going to move it to a less active form. And, very importantly, it's going to dephosphorylate glycogen synthase as well, which causes glycogen synthase to become active, which is now going to start taking all that glucose and making glycogen again. You betcha. You betcha. You understand what's going on with that? I mean, there's a dynamic. There's a dynamic that's here. It is. It is. It's a very good question. But as I said, your questions are exactly to the heart of this type of regulation. But this type of regulation allows us to respond to the fright situation, the glucose poison situation, and the sitting around doing nothing situation. Because for the most part, if I'm sitting around doing nothing, I'm not producing epinephrine. I'm not really producing insulin. And so there, it's the up or down of this, or the up or down of this, that's really governing everything. So having that up or down control in addition to the hormone control is really, really beneficial. Okay. So our question is, if I go from phosphorylase A to phosphorylase B, is it going to be in the R form or the T form? It's probably going to be in both. Okay. But let's imagine the situation that I've been describing to you. Let's imagine a situation where the cell is, I'm not exercising, I'm not burning that energy off. When my insulin kicks in, it's going to go this way. And if my cell has plenty of energy, what's the level of ATP it's going to have? It's going to be high. So it's going to go to the T form. Right? So if I'm sitting around doing nothing, this guy is going to tend to move to the left and upwards. If I'm exercising, it's going to tend to move to the right and downwards. Make sense? Because on the left, moving upwards is going to happen under conditions of high energy. ATP, glucose 6-phosphate, both are present when I have high energy. Glucose is present when I have high energy. Only when I have low energy, AMP, do I see T go to R. Over here, once it's in this form, it automatically flips down into here. Okay? Now, there's 4 different things to think about there, but they all do relate to things that are happening inside of your body. Different circumstances. Say it again.
Why do we have to have for the glucose but not for ATP and AMP?
Well, this is a different, her question is, why does this one respond to glucose and this one doesn't, right? Is that your question? Okay. Well, these are different forms of the enzyme. So because they have different forms, slightly different forms will have slightly different shapes and slightly different ways of binding things. So this guy, because it's got the phosphates on there, is able, well actually, it doesn't bind glucose, it only binds glucose when it's down here. All right? This guy is able to bind glucose and convert it back to this form. Right? That can't happen over here because there's no phosphates and the enzyme isn't in the right shape to bind a glucose. It's in the right shape to bind an AMP, or it's in the right shape to bind an ATP or glucose 6-phosphate. See, this is why I wish I had taught you guys protein structure, because understanding protein structure really tells you how very tiny changes in protein structure change how enzymes bind things, change how hemoglobin is affected by oxygen and things like that. Okay, I'm not expecting that you're going to, I'm not going to ask you about that because I didn't teach you that. But that's where it would have been beneficial if I had. Yeah. Does cyclic AMP play a role in this? The answer is no, it does not. Just AMP. Just AMP. Yeah. Okay. All right. So good question. Let me just answer your question, and I understand your confusion. So I've talked about cyclic AMP with respect to the signaling process that ultimately leads to this. This, as you see on the screen, does not involve cyclic AMP. All right? But getting to this point actually does. All right? So since you've asked the question, I'll ask it. All right. Actually, let me show you here. It's actually something I talked about today. You won't be responsible for this, but I'll show you it's because you've asked the question. If we look from today's lecture at this guy right here, all right? This actually shows the process that's involved not only in the breakdown of fats, but also in the control of glycogen metabolism. Epinephrine binds. It stimulates the production of cyclic AMP. That's something I told you about relative to caffeine. Cyclic AMP activates a protein kinase, which puts a phosphate onto things. One of the things that ultimately gets a phosphate put onto it is glycogen phosphorylase. Okay? So cyclic AMP plays a role in that signaling process, but it doesn't play a role directly in the glycogen phosphorylase part. Okay? Well, you will need to know about cyclic AMP where I talk specifically about cyclic AMP. So where did I talk specifically about cyclic AMP? I talked about cyclic AMP with respect to caffeine. Okay? So if I, oh, come on. I showed you this figure. I said cyclic AMP, all right, is necessary for signaling to make glycogen phosphorylase with a phosphate on it. All right? So the signaling process makes cyclic AMP, and cyclic AMP helps ultimately to be sure that there's a phosphate on there. Okay? As long as cyclic AMP is present in the cell, enzymes will get phosphorylated. Okay? Well, normally the cell has a protection against that. It has an enzyme called a phosphodiesterase that routinely breaks down cyclic AMP. So if epinephrine comes along and stimulates cyclic AMP, the phosphodiesterase starts binding to it and breaking it down, and so cyclic AMP concentrations start to fall. So normally cyclic AMP concentrations go up, they activate the enzyme, and then they start to fall. Right? Okay? When you drink caffeine, phosphodiesterase gets inhibited. So the cyclic AMP concentrations go up, but they don't really start to fall, at least not nearly as fast, because the enzyme is inhibited. And as a consequence of that, this form of the enzyme right here predominates, which means that your body is breaking down more glycogen and releasing more glucose to the bloodstream than it normally does. That's why you get a buzz from caffeine. Does that make sense? Okay. Yeah? Yeah, what's cDNA? All right. So cDNA stands for complementary DNA, and it's made from RNA. All right. So that goes back a ways. Let's see here. So when we talked about biotechnology, wrong one. That was here. Okay. So when I talked about technology, I talked about an enzyme called reverse transcriptase. Do you remember what reverse transcriptase does? Anybody? Makes DNA from RNA. So if I wanted to make a copy of a messenger RNA, all right, and let's say I had eukaryotic cell. Well, in a eukaryotic cell, all the messenger RNAs have a poly-A tail. So I can make a primer that consisted of a bunch of T's. A bunch of T's, right? So I'd take this messenger RNA, I'd put this T primer, this multiple T primer on there, and then I would add reverse transcriptase, 4 DNTPs, and I would make a copy of that messenger RNA in DNA. That's what I would do. Okay? That copy that I made would be a cDNA. C stands for complementary. It's a complement to the RNA that it was made from. Make sense? It turns out to be very useful for making, in the laboratory, copies of messenger RNAs that happen. So you remember the microarrays that I talked about, where I said we copied all the messenger RNAs of a given cell? We would use a reverse transcriptase to do that. Yes, back in the back. Okay. The question is, can I talk about how Pfk is activated by F26BP? There's not really much to say other than the fact that it is. The F26BP is a reciprocal regulator of Pfk and the corresponding enzyme in gluconeogenesis, which is F16BPase. Okay, or F16 bisphosphatase. It's a very important reciprocal regulator. It's probably the most important thing that actually controls those two enzymes. I talked about, for example, ATP and AMP affecting Pfk, but they really only affect Pfk under fairly high concentrations, and it takes quite a bit to affect those enzymes. F26BP affects it in very tiny concentrations. In fact, it was many years before people even discovered F26BP because it's present in such tiny amounts in cells that they overlooked it. Okay? But the important thing is that F26BP activates Pfk, which activates glycolysis, and at the same time it inhibits fructose 1,6 bisphosphatase. If you take it away, the reverse happens. That is, the F16BPase becomes active and the Pfk becomes inactive. And there's a long, convoluted story about how F26BP is made, but like I said, I'm not going to tell you about that, 'cause that gets more than we need to do in this class. You guys have enough to know. Why we don't have what? Oh, the glyoxylate cycle. Okay. So do you need to know why humans don't have the glyoxylate cycle? Well, I guess I would say, did I talk about that? So what I said was it was unfortunate that we didn't have the glyoxylate cycle because if we had the glyoxylate cycle, we would have a way of making glucose directly from fat, and we can burn up glucose pretty readily, so it would be a very nice cycle to have. The reason we don't have the cycle is because we don't have the cycle. I mean, and that's actually a true story. I mean, there's not really a reason per se. We don't have the cycle because we lack the two enzymes that the plants and bacteria have. They have one enzyme is called isocitrate lyase, and the other enzyme is called malate synthase. We lack those two enzymes, so that's why we don't have the cycle. Everything is clear as mud then, huh? Yeah, no, I hear you. And that's one of the disadvantages of doing the review session early. I've got to go to Portland tomorrow night to catch a plane Thursday morning, so I figured if I didn't do it tonight, I wasn't going to be able to do a review session, so that's why I'm here tonight. So it does help with you looking at this over time. Did I give you guys my cell phone number? Okay. So you're welcome to give me a call. I mean, I carry my cell phone all the time, so if I can answer your call, I'm happy to try to answer your question for you. Yeah, I'm going to be in Boston, so I'm going to a meeting. So just give me a call. Yeah, Olivia. Oh, okay. So the question is, what's the advantage of the glyoxylate cycle with respect to not releasing carbon dioxide? Okay. The advantage is that if you look, let me just show you the cycle. Okay. So if you look at the glyoxylate cycle and you compare it to the citric acid cycle, right? In the citric acid cycle, you start with 4 carbons and you end up with 4 carbons. In the glyoxylate cycle, you start with 4 carbons and you end up with 8 carbons. And so it's right here, okay? So ignore the, this middle reaction. Let's just say we're talking about the citric acid cycle going around here. Citric acid cycle starts with oxaloacetate, acetyl-CoA. We put 2 carbons in, and we lose 2 carbons through decarboxylation. That's why we start with 4, we end with 4. All right? In the glyoxylate cycle, we start with 4, we add 2, we don't lose any, and then we add 2 more. So that's how we start with 4 and end up with 8. The advantage of that is that every time the cycle turns, it spits off another 4-carbon molecule. And the 4-carbon molecule it's throwing off is oxaloacetate. So that's why plants and bacteria can make glucose in net amounts from acetyl-CoA, and we can't. The reason we can't is if we start taking away our oxaloacetate, we stop the citric acid cycle. So we could make a little bit of glucose from the small amount of oxaloacetate that's there, but it would stop the citric acid cycle. So we can't make glucose in net amounts from acetyl-CoA. But plants and bacteria can do it because they don't lose those carbons to decarboxylation. That's the reason why the bypassing those reactions is important. Make sense? Oncogenes? Let me just say one more thing, then I'll answer your question. When I talk about photosynthesis next week, you're going to see that glyoxylate is produced as a byproduct of photosynthesis. And glyoxylate turns out to be a toxic substance. Okay? So we'll see how that plays into photosynthesis. And this is one of the reasons that plants actually have this cycle. Okay? No, I'm sorry. Oncogenes. Okay. So the question is, are oncogenes found in normal cells or only in mutated cells? This is a semantic question, all right? Meaning that the word itself has meaning. All right? A normal cell, by definition, will not have oncogenes. It will have proto-oncogenes. Okay? Because proto-oncogenes are the unmutated forms of these genes that function normally inside of cells. So you have RAS. You have other proto-oncogenes that can become mutated and give cancer. But it's the mutated form, and you will find the mutated form only in cancer cells. And when it's mutated, it's called an oncogene. Yes? Okay, so the question was, I asked you to know structures of glucose, fructose, ribose. Did I ask you to know the structures of anything else? I think I relieved you guys of having to know sucrose, which I usually make people learn. And you don't have to know lactose either, no. So glucose, fructose, ribose. That's the 3 most common sugars that you're going to find. And you will need to know those structures in other classes. So I think those are reasonable. Yes? You said sucrose? So that's a good question also. Professors like that as a trick question. All right? Sucrose can't exist in a Fischer projection because the anomeric carbons are tied up. Yeah. But I didn't ask you to memorize the structure of sucrose. Yeah. But you do need to know the Fischer and Hayworth forms of both glucose, fructose, and ribose, right? Not both, all 3, yeah. whatever the word is for all three. What's that? Yep. Can what? How that works out? Okay. Well, let's go to the reaction. So that's a decarboxylation reaction. Here you go. This is the thing down here with all these various arrows, right? That's what you're confused about. Okay, don't be too confused. Let me just show you what's happening, all right? So let's do the simple things first. The simple things are that carbon dioxide is being released because it's going from 5 carbons to 4 carbons. Okay, so carbon dioxide has to be released. This is an oxidation, and so as an oxidation, NAD is being converted into NADH. Okay, and the last thing that has to happen is a CoA has to get put onto here, and that's what that's showing right here, is the CoA's coming in. It's just showing it as a circle because it's released after that, and it becomes released up there. Does that make sense? Yeah. So there's 3 things that's happening there. It's the energy of that oxidation that is helping to make, or helping to link that CoA to the succinate, and that turns out to be an activated intermediate. And an activated intermediate, in this case, is using energy to make GTP. Yeah. It comes off right after. So it goes on, and then on the next step, it's lost. See, it's going on in this reaction. It's coming off in this reaction. Yep. Notice that these, okay, so here's this, you don't need to know this, but it's relative to something I said today, all right? So I said today that fatty acids are going to denature proteins, right? If you think about the structure of a fatty acid, why do they denature a protein? They denature a protein because one end is nonpolar and the other end is polar. So consequently, they can, the nonpolar portions of them can interact with the nonpolar part of a protein. Did Dr. McFadden talk about how the nonpolar parts of a protein are on the inside of a protein? He did? Okay. All right. So that's true. The nonpolar parts of a protein are generally found in the inside part of a protein. So if you have something nonpolar like the end of a fatty acid, you stick it into a protein, now it's got to unravel a little bit. Right? And so that's why detergents denature proteins, is they cause that nonpolar part to sort of unwind, all right, and start interacting with more and more of those soap molecules. Right? Why do I tell you that here? I tell you that here because you say, oh, well, the CoA is going on there to protect this thing so it's not denaturing proteins. Then why does it come off? Well, CoA goes on sometimes for reasons other than protecting proteins, and this is one of those cases. This molecule right here, right, when the CoA comes off, you see there's its structure. Carboxyl, carbon, carbon, carboxyl. There's no end of it that can stick into anything because both ends are polar. Right? If this were COO minus and that was a CCC, that would be more of a problem. Make sense? A little tidbit. You don't need to know that, but that's a little tidbit. I like to share little tidbits every now and then. These things are cool. You know, I mean, biochemistry is I talked to, in fact, I'll tell you a cute story. I talked to a former student of mine who took this class, it's probably been about 7 or 8 years ago. This weekend, she called me up, and she says, I'm thinking about coming back to school. And I said, really? I said, what do you want to study? And she says, well, I'm thinking about biochemistry because she had so much fun taking biochemistry. And when she was here, she didn't think biochemistry was that much fun, but the more she thought about it, the more fun it seems. So biochemistry is fun. It's true. When I took biochemistry myself the first time, I hated biochemistry. I thought it was the most god-awful thing on the face of the earth. I said, this is a true story. I had to take one semester to do it on a semester basis, and I got out of the semester of biochemistry, and I said, never again am I going to do that again. Look where I am. You know, the bottom line is never say never, right? Okay, we're boring the people out in TV land, so I should shut up. Anything else? Clear as mud? Like I said, if you have a question, give me a holler. I'll be happy to try to answer it for you if I'm free and not busy. And good luck on the exam. I'll be here tomorrow as well, so I'll be lecturing tomorrow.